Hi everyone, welcome to this month's Bee Biosecurity online talk. My name's Rebecca, I'm the Bee Biosecurity Officer for Queensland, and we have a really exciting talk tonight. Before we get into that, just a quick reminder, we've got one more talk in this series on the 6th of April, same time, 7 o'clock. And this is going to be on swarms and catching swarms. Now, I'm going to put the same call out I did last time, is that if we do have a beekeeper in the area and they do have a swarm they're going to catch and they wouldn't mind me filming it, I would absolutely love it. So give me a call if you have a local swarm in Brisbane that you're going to go and catch and wouldn't mind some a little bit of footage taken of it. So tonight I'm joined by a whole suite of people, but the key person is our special guest, Professor Stephen Martin from the UK, who's going to give us a really interesting talk about some of his experience over there, particularly with the impacts of Varroa, Deformed wind virus, and Hornets as well, with a little extra added bonus. We've got a couple of people helping us this evening with production as well, of course. We've got Susie and Kerry, and we've got Matthew Cook as well. And we've got a few people online who are going to help us with some of the questions. Of course, our apiary officer, Hamish, and hopefully we're also going to have Rob Stevens from the National Varroa Eradication Program, if you have any questions that are a little bit more local to Queensland at the end. So without further ado, I'm going to throw it over to Professor Martin to get us started.
Right, well, thanks very much for inviting me to this talk this evening in Australia and a very cold and murky morning in the UK, where we're still under our third COVID lockdown. So it's very, very different, chalk and cheese. So what I'm going to talk about tonight, I've been working on Varroa and long-term solutions to Varroa, are just understanding Varroa and actually Hornets for about 30 years of my life, pretty much all my research career, except for the first 7 years, which was spent mainly in Japan studying Hornets. And recently that work has been in much demand. I'll bring you right up to date with what we're actually doing, what our latest thinking is. And for the first time in, I'd say about 30 years, the last few years, we can see a light at the end of the tunnel, and I'll go on to explain that. So obviously I've got 30 years of work to cram into this little lecture, so I've really shortened things down. But there's all the main players on the first slide. All right, let's see if this works. Okay, so it's a bit strange giving a talk to people in Australia because the majority of them have never actually seen one of these things. And so this is the Varroa Mite. We've had it for 30 plus years now. It's worldwide. You are one of the very few countries in the world that don't have it, or at least the destructive form, and long may it be, because it has been incredibly devastating wherever it's got. So I'll run a little bit of basic biology. So it lives entirely on honeybees. It has two parts of its life phase. So most of the time is spent on the adult bee. It's very rare you'll see it like this. They're normally underside the underside of adult bees, tucked in between the plates and very difficult to see or remove. And then they have their reproductive phase, and they reproduce in the brood, and they're highly specialized at this. The dark brown mite is the foundress mother. Then in between the abdomen and the thorax, the smaller one with the darker legs, that's a single male. It's always a male produced first. And then the other odd ones that you can see, that's another young mother. It's just hatched. And the little white skin that's sitting at the bottom of the cell, well, on the actual cell capping, you can see that's a shedded skin, and that's how we know that that's actually another mother. It's another fountain now going to be, it's got 2 females in there, even though they look very different colour, and they will darken over time. So that's its life history. And most beekeepers have thought, it's just a small mite, so, you know, do we have to really worry about this? But actually, this is the result of millions of years of evolution. It is highly evolved to live only in honeybees and only effectively one species of honeybee. Its shape is unusual for a mite. The curvature of the cuticle of the skin, the outer, is perfect to a bee. It slides perfectly under. It has piercing mouth parts to be able to feed on the bee fat and some of the bee hemolymph. Its surface is chemically identical. It's camouflaged, so bees can't see it. They can touch it, but they just think it's another lumpy bit of a bee. My favourite is a things called Peritreams. These are little snorkels that they pop out when they go into the brood food, where they lay quietly before the cells are capped over, and they breathe through the brood food. They've got retractable lobes, they've got claws, they've got hairs to be able to hang on to the bee. So they're incredibly highly specialised. And most beekeepers have made the mistake thinking, well, our bees, they'll outsmart them. And they haven't till quite recently. So it is a formidable foe. And outside Australia, it remains, even after decades of being present and being treated, the #1 enemy of beekeepers, or the #1 concern. They're still having to treat for it in the vast majority of the northern hemisphere. And you can see the impact it's had in New Zealand when it arrived there. These are all the life stages. Most beekeepers never see these, but just to show you how specialised they are, they give birth to it's not an egg, it's actually a larva, very large, and then they either grow up into females, the top row, or actually they have a single male on the bottom row, and it's brother-sister mating. Lots of mites do this. This is not unusual, and they like to keep the reduction of genetic variation by brother-sister mating is highly valuable for them, because they live in a very invariant world inside a bee colony, be it in Australia or Alaska or in Brazil. It's the same temperature, it's the same constant, and so the mites are highly adapted to these. We know so much about them. We're a huge amount, and this is some work that I did 25 years ago, and it's still being used today. There was a couple of us did this, and we worked out the mite reproduction and how many offspring could be born at the end, but we know a lot about it. And it turns out that Varroa reproduction is the key. You need lots of mites for it to kill a colony. So its original host, as probably most beekeepers in Australia will know, is Apis cerana. It was thought to have jumped the species barrier once, but we know now it's actually done it a couple of times, and it's generated a couple of mite species. But the one we have to worry about really is the destructor that spread very successfully around the world. And Apis cerana is very highly adapted to these mites, and it can only the mites can only breed successfully in the drone brood. They avoid the worker brood. It's still not 100% clear. It's a bit controversial, the reasons why they do that. But the drone brood is only present potentially a few times of the year. The numbers are very small, and also they have a density dependent system. So that means once you get multiple mites going at Apis cerana drone cell, they will kill the drone brood, it will die, and then they just seal it up. The bees entomb it, and so the mites don't emerge. And this means that you never get many mites in an Apis cerana colony, but they all have them. Pretty much every colony you look at have a small number. So they keep it way under the economic threshold. When it moved over to Apis cerana, this is where the problem started. So when I started researching this, it was a very clear picture, and that was one of lots of mites, vampire bugs, they used to call them. They drink the blood, or now we know it's actually the fat, of the developing pupae. They cause it to shrivel up and die. And so it was just the simple weight of mites that was killing colonies. A very simple picture, when I say. And so we did a very simple experiment. My job was to work out how many mites could kill a colony. And one of the first experiments I ever did was we just left some colonies. Each bar is a colony. And we looked at the total mite drop, and we looked at whether colonies survived or died. And the problem that we came up with was, well, there was no difference. It was irrespective of how many mites you had in a colony, of whether it died or survived. And we couldn't understand this, and we tried to publish it, and we couldn't get this published because they said you didn't know what you're looking at. And so this raised alarm bells. So I wasn't really convinced with what we really understood what was going on. And then I had a very big stroke look. At the time, I was working for the government in their national bee program, studying Varroa. And we were very fortunate in England to have a honeybee virologist, and that was Brenda Ball. And the only people studying honeybee virology was Brenda Ball from the UK and Dennis Anderson, your very own, from Australia. So they would literally well, there was 3 people and Bill Bailey who originally started all this. So I got together with Brenda because Brenda thought that maybe the viruses had something to do with this, but she was never sure. So we repeated this experiment, bigger scale, to see what happened. And what we found was where we couldn't see any viruses. So this was, these are the colonies in yellow. They all survived, but their mite populations could be extremely large. However, then when we found colonies that survived where we did detect virus, actually the mite populations are really small, and which are the green bars. And then when it was red, these are colonies that collapsed with different numbers of mites in, and they had a virus. At that time, we were testing a range of viruses we didn't know what we were looking for. And then we'd cracked it. We knew there was something else going on. And then what we did, it was a long story short. In the end, we identified really it was deformed wing virus was the single most important virus for a whole bunch of reasons. Let's go on. And the main reason for that was that deformed wing virus did not kill the pupae, and it didn't kill the adults immediately. It allowed them to live for a certain amount of time. And we came up with this scheme a long, long time ago from that original work. And number 1 was hypothetical at the time, which was basically you have a mite with no virus and it just lives in a bee colony. And we always believed that the situation would sort itself out, a resistance would arise. To kill a colony by drinking or feeding on the fat, we estimated you need about 80,000 mites, and we'd never seen that number. However, when these viruses appear, and they are very rare in the population, so currently in Australia they've not been detected, but you can't detect every single colony. And we've looked in places like Hawaii where there's no Varroa, and we were finding about 5%, which is quite high, but at very low levels. But occasionally we know it can replicate up and burst out and kill a colony. If a mite is in that colony, when the colony dies, it doesn't actually die, it collapses. And that means the bees and the mites move out to other colonies as drifting. And then once they get into it, once they've got the virus, they set a new transmission cycle up, which is in number 3. And then what happens then is they transmit it to the pupae during reproduction, but it doesn't kill the pupae, because if it did, that mean that the mites couldn't reproduce, the vector would die, and the whole system would crash down. And that's why more virulent viruses like acute paralysis virus or slow paralysis virus that are really lethal to the pupae, the mites, although they do transmit them, the populations soon die out because the mites can't reproduce. The pupae then emerge as adults. A very small number of them are deformed. That deformity is caused by where the mites feed, and it's quite unusual to feed on the wing buds, and if that happens you get deformed wings because the virus replicates in the developing deformed wings. But for the vast majority of them, the virus grows up in the emerging bee, and then it has a reduced lifespan of it lives about 1/3 of its life normally, but 2/3 of its life it dies prematurely. When it goes into adult bees, it can be transmitted into the adult bees, but if they've emerged from a pupa that's not been infected, the adult bees are okay. They change their foraging a little bit. The impact is not as great, and they can survive and act as carriers. So basically, it's a combination between the two. And the big breakthrough we had, it's a little bit complicated, but I'll just, I suppose with the world of COVID now, everybody's a little bit more knows what's going on. So this is some work we did in Hawaii after the mites first came. And all I want you to take away from this picture is that deformed wing virus isn't a single strain or variant. It's actually hundreds of variants. And in populations where you don't have Varroa, or Varroa has just arrived, you have a very diverse population, and that's indicated by all these wiggly coloured lines. Each one is a variant. Once Varroa becomes established, within a few years, you get total prevalence, which is the red pie, and then the red bars on Oahu, 2009, you just end up with one strain. And this is exactly what COVID is doing. We've just had a strain in the UK that was arisen in the south of the UK, and it spread right through the UK very fast, very transmissible, and now is going around the world. So these viruses are constantly changing, trying to get better at transmitting. So the big thing is it takes a few years, and this is important for people in Australia. From when the mites arrive, there's usually a lag of about sort of 2 to 4 years when everything's fine. The mites are there, their numbers are building up, nothing's really happening. And it's when they start to pick up the virus from wherever they get it from, and that's never sort of certain. They thought it may have come in sperm in New Zealand. And then you lose this, because of the new transmission cycle, you lose the strain diversity, and the prevalence goes up, the load goes up, and then the colonies start to die. And then you're locked into this permanent situation where you've got to treat. So what you've got are two sides of the same coin. One side is deformed wing virus, and the other side is Varroa reproduction, and you need both initially to kill colonies. Although, as I say, it takes 2 to 3 years, and that's the period where beekeepers are always getting really confused because nothing's actually happening on the ground. And then suddenly it happens really fast once the virus starts to spread very quickly as colonies start to collapse. And now, from something that was a benign, extremely rare virus, only discovered, it was the last of the main bee virus to be discovered. Now it's the most common insect virus in the world due to the worldwide spread of bees. It's been detected in 55% of all colonies or apiaries across the 32 countries that have actually done surveys. It's not just a honeybee virus. It's in fact, we've been detected in over 65 non-species, non-honeybee species across 6 orders of insects and mites. So it's a very widespread, particularly in the Hymenoptera, bees, wasps. You can find it. We don't think yet that it causes a lot of impact in many of these other species. There is some evidence for bumblebees, but for a lot of the other things like ants, it is present, but it's actually, the load is very low and it's probably still quite diverse. And currently there's a very big survey, it's not actually been detected in Australia. We say not detected because you can never prove a negative. So it may not be there, but it is possible if you look hard enough. But there's nothing you can do about that. And what we've been doing over the last few years, we've been tracking the changes because as COVID did, deformed wing virus is changing. The original strain was the A strain. We now call that the red one. That spread throughout the world and killed most of the colonies. Now it is changing into the B strain and being replaced by it. This is the blue strain. This has become much better adapted to the system, where a deformed wing virus A cannot replicate in Varroa mites. Deformed wing virus B can. So actually it's becoming better transmitter. And then we have this really odd deformed wing virus C. We rarely see it. It pops up here and there. We think it may be very lethal, and hence we don't see it very much, but it's always there, but in very, very low amounts. And in different countries, it's at different stages. So in the UK, we've pretty much transitioned right over to deformed wing virus B now, as is most of Europe. So what if when you get the Varroa and eventually get the virus, what strain you'll get? It's not really any different to the beekeeping because both are as bad as each other, we think at the moment, but it's just the virus getting better. So we're going to ask the question because we can now address this question. What would happen if there was no deformed wing virus? And this could be a situation that you're faced with at some time in the future. So I was very fortunate, I was working in Brazil, to go out to this small island about 350 kilometers off the coast of Brazil called Fernando Noronha. And on that island is a European honeybee population, and it's the longest naturally resistant population to Varroa in the world. It's getting to 30 years now, and it's never been treated. And there's plenty of Varroa there. And it's an accident. They thought they'd set up a queen rearing station on this remote island so they could then feed queens back to get rid of the Africanized honeybees. Obviously that never worked. Africanized honeybees were way too successful and they spread throughout Latin America. And the reason they were successful was that they were resistant to Varroa. So that was one of their big competitive advantages. So we were fortunate a few years to get out to this island and try and understand what was going on. So they've had constant mite levels for about 25 years. They've had no losses, and when we had a look, basically deformed wing virus is absent. It's just not present in these populations. And very interestingly, mite resistance has appeared. So the reproduction in the mite population on this island is just very low. And we actually calculated it from observations, and we worked out that they produce about half a new offspring that's mated per reproductive cycle, and they do between 2 and 3 reproductive cycles in their life, so they can just about hang on. In fact, the worker brood infestation level, it averaged about 20%, and it's stabilized at that for a long time now. And about 40% is maximum. It's about 35% in the drone brood. It hasn't gone up, it hasn't gone down. And most of these are non-fertile mites, so they're just trying to reproduce and they can't, they've run out of eggs. So this is a possibility what happens. We don't know the precise mechanism in these. Unfortunately, I can't get back to work that out. And now we know that actually this is not a unique situation. There's no deformed wing in Solomon Islands, and we've got the same situation. And then in Papua New Guinea, the Apis mellifera colonies there, they also don't have deformed wing virus. Slightly complicated here because this is a race of Varroa jacobsoni that's been able to jump into Apis mellifera colonies. So this is another case where they've jumped over, but they've pretty much stayed the same, and these are in Papua New Guinea. And this is all the work done by Dennis Anderson. So we've started looking at the long-term evolution of Varroa resistance. I've been doing this actually for a long time, and we worked out down in Mexico and Brazil and actually South Africa. But this long-term evolution, so basically where people don't treat, has been very common in some countries, basically because they couldn't afford to treat. Northern hemispheres have always treated, but in the southern hemisphere they couldn't afford, and the bees quite quickly became resistant. And we were interested in understanding the mechanism behind that. And it turns out that now that there are populations springing up all throughout Europe and the United States of individual beekeepers or groups of beekeepers that haven't treated for over 10 years now. So there is also resistance appearing in Europe, and that's why we started sort of reinvigorating our research. And we know that now, we now quite understand it quite well what the mechanism is. And it's to do with the virus, the mites, obviously the honeybees, because there's loads of honeybees around the world, beekeepers, there's loads of beekeepers around the world, and then there's a very well-known behavior called hygienic behavior with bees that perform. And you need to have all these, you need to understand them all to be able to have any chance of getting long-term Varroa resistance. And the reason being for that is if you say you bred a bee, which people have tried to do, then, you know, beekeepers go, oh, no, I want that type of bee, and oh, I want that type of bee, and no, I don't want a resistant type of bee, no, but I need this type of bee. So people, you know, beekeepers are funny people by their very nature. Everywhere around the world are the same. And then we've got all these different types of bees. So we've got all these strands to put together. The key is reproduction. So we've already known this for sort of nearly 20 years now. And this is very stylized, but reproduction in European colonies, it's basically Apis mellifera, is that you get one mite comes in and two mites come out. That's just two offspring come out, and then the population just increases. And if you don't, you treat it. In resistance colonies, they have a lower level of reproduction, not like on the island where it's 0.5, but it's something like about 0.7, while in England it's like 1 point something, 1.4. So it's that what causes that reproduction to be lower. And one thing that the beekeepers had noticed, well, throughout Europe, was an increase in ball brood. Now, you should not see this very often in Australia. Occasionally it happens genetically. And this is where you get cells that are completely opened, and they have sort of white, pink-eyed pupae, and they're always the same age pupae, just sitting in the bottom of the cells. And nobody really seems to know what happens to these cells. Some can get removed, but actually some can get resealed over. This one's just been done, so they actually will have a look inside and then reseal it over. This cell here actually has mites in it. You can see the mites on the wall. And we hadn't put together this behavior and bee resistance. And so what's happened recently is that we've been able to put things together due to a single person. There was a big breakthrough study in a couple of years ago by Melissa Odey. She's a Scandinavian PhD researcher, and she was able to associate a trait called cell recapping with tolerance or resistance. And we've known that hygienic behavior is a very common trait in honeybees. And it's really useful for them to detect diseases, maybe like wax moth or foul broods, chalk broods, and then they detect them and they remove the dead pupae. The problem with Varroa is that it doesn't kill the pupae, so it doesn't actually, you're not looking at a dead individual. And we've realized it takes a fair amount of time and selection, which is removed when you start treating, for them to learn to adapt to this new odor and being able to differentiate between cells with Varroa and cells without Varroa. And what we realize now that bees that are resistant are able to detect this smell, make that differential. They open up the cells, which they investigate the cells by uncapping. And if it's an infested cell, they tend to uncap it larger. If it's a non-infested cell nearby, they'll put a smaller hole in it. They'll investigate it. And then they have a choice. They can either recap the cells, and that's like put more wax on top of it. And they do recap cells with mites in because the mechanism's not perfect. Or they'll decide that this is an infested pupae and they remove it. And they remove it by cannibalism, so they'll eat it. The mites, as I said, are very highly adapted and quick, and the mites escape. So the bees don't catch the mites escape, but they haven't reproduced. They've wasted all their eggs, and they only have a limited number of eggs, around about sort of 18 to 25 eggs. So they're going to have to go and try and find somewhere else to reproduce again. This is a trait that was linked with hygienic behavior, well, linked with the resistant colonies, and we'd all overlooked till a few years ago, just 3 years ago now. And these are just the normal cell caps. If you turn them over, so you invert them, have a look on the underside, you'll see this one is the top right-hand one is just a silk cap, and that's completely natural. That's how nearly all the ones in Australia will be. It's absolutely just normal. But then what you can find sometimes are ones with the center that's been cut out, and there's a small area in the middle that's not silvery, it's dark matte, and that's because there's a hole been made and it's been refilled in with wax. The wax hardens on the outside, and so the color just becomes uniform, but on the inside you can tell. And so here there's a much larger in the lower right hand panel. And that's what we called a recapped cell. And what was worked out in places like Australia, we have some small islands like Colonsay and the Isle of Man. These are places where we don't have any Varroa. Then this recapping, the percentage of cells that are recapped, and clearly they're yellow bars, which means they're non-infested cells, is very low. It's often very hard to tell. These are population means, so these are several colonies, not one colony. But then in susceptible colonies, so these are treated colonies. Each 2 bars represent a population from Norway, Sweden, 2 from France and from the UK. And what you can see is they do detect a small number, they do recap a small number of non-infested cells and a few more infested cells, but they don't do it very well. So because the red bars are bigger than the yellow bars, it means that bees can detect Varroa, and that's exciting. But it's just the susceptible populations are really bad at it. We go to Varroa resistant populations. We've got some in the UK, a couple in France, 1 in Sweden, 1 in Norway. We looked in Brazil, and then 2 races of bees in South Africa, the Scutellata and the Capiensis bees. And they were really good at it. You know, they're hitting sort of the Capiensis bees hitting about 80% of all infested cells. They're opening and at least having a look at. And they're usually removing these eventually. This opening and closing is a very complex behavior, and Marla Spivak's done a lot of work in Minnesota on this. It's highly complex, and this is what results in this variability. But long story short, this is what reduces the reproduction of the mites. Forget the complexity of this curve. It's just that where this number here, the population decreases in the bottom part of the curve. This is the mite population, and the population increases at the top part of the curve. We've been able to calculate that 40%, if you can get 40% removal by your bees, and that will generate a recapping rate of sort of 60 to 70%, so quite high, then the population of mites decrease. So we know that this is how this system is now working. And we put it all together now. So increased mite detection, which has now actually been worked out what that is, leads to increased brood removal, infested brood. Recapping is, we think, is a proxy, although it may have an effect. Quite a relationship between these two is not 100% certain worked out. But whatever, increased recapping and increased brood removal lowers mite reproduction. That decreases the mite and the virus burden because you've got less vectors, and then that increases your colony survival. So this is what happens in resistant populations. And for example, in the UK, we're currently doing a survey, and there's about 10% of the people haven't treated for over 5, actually most of them 10 years, and about 20% of people are not treating, have started not to treat. And we've got one area, a huge location in North Wales. We've got about 100 beekeepers and about 500 colonies. It's quite a remote national park sort of area, and they haven't treated for 10 years. So there is a big increase, and I think in the next 10 years in the UK, the majority of people won't be treating for Varroa. That they'll basically be using either splits or queens, and we're still working on the best way to achieve that. We've actually produced a little booklet for the British beekeepers, which outlines a lot of what I'm saying a lot more, and some techniques for doing these things. It's like a pint of a beer, so it's quite good for our British Beekeeping Association. And finally, I just want to because I've worked on this, just to bring you an alert about the hornets. So The world distribution of hornets now, naturally, they're northern hemisphere species, but they are invading, and we've had some real problems in Europe and actually Korea and now Japan of a thing called the Asian Hornet, which is this fellow here, a highly specialized hornet, sometimes called the yellow-legged hornet, which is the best name for it because it's very distinctive. In Europe, we call it the Asian Hornet, which is very confusing. It's Vespa volutina that its name. It arrived in 2004, we think in Chinese pots, we're not sure, and it spread incredibly rapidly throughout Europe. It's now into Portugal, it's in Spain, it's throughout most of Europe, and it's predicted to spread pretty much throughout a lot of Europe. This is causing vast problems for beekeepers because these things are very, very good at hawking bees. It's very close to your shores. So this is a natural distribution of this particular species, and it's right down to Borneo Flores. The main problems is this Nigerithorax subspecies from central China. This seems to be quite an invasive. This is the same subspecies that's in Korea and also now moved into China. So this is something that's big, it's easy to look at. We've been eradicating it in the UK successfully for several years now, although it's costing a lot of money and a lot of effort to do this. We're getting better methods at the moment, but these are becoming very good at moving around. And the big problem here is this particular species is really likes urban areas, very highly adapted. And basically in urban areas, it's eating about 50–60% of its food is honeybees. Forestry area, it's only about a third. Honeybees allow it to live longer, and I think it would clearly do very well in Australia if it ever reached your shores. And this is its classic. It's a real master at catching honeybees. That's what it does in its native range. It hawks, it hovers outside the colony and catches incoming bees. And it's extremely good. It's the best species of hornet in the world, the most agile for actually catching honeybees on the wing. And because our European honeybees or Western honeybees have no defences against it, then they just get caught far too easily. The nests can get extremely large. These are mature hornet colonies, and so they can have a devastating effect on the local bee yards. And now, surprisingly, is the giant Japanese hornet. There's been 2 invasions of this separately, one from Japan and one from Korea. These are the world's largest hornet. The Americans have called them murder hornets because they murdered the honeybee colonies. These are extremely large. These are Matsuono's pictures. Quite common in Japan. They plunder Apis mellifera colonies. This is the classic. They can do this within 2 or 3 hours. I've seen this happening. And they're not interested in the bees, they're interested in the brood. They do these mass attacks, and these have already been seen. They've already been seen up in the Vancouver area and down just inside America. It's well established there. They don't admit it, but it's very highly spread around, and it's all forest in that area, so it's perfect area for them. So I want to finish there. I thank everybody for listening. All the beekeepers that help me with the work worldwide. The funders again from all over the place. And then this is my team, or part of my team over the last few years, that have been working on bee viruses and bee Varroa. And thank you very much for listening. And I'll end my show there. Thank you.
Thank you so much. That was really interesting, and I have learnt so many things, and I think it's really some key takeaway messages tonight for a lot of our beekeepers here in Australia. Particularly, I think when you started by saying you've been working for 30 years and you're just seeing the light at the end of the tunnel, I think that's something that we can really take away, in that, you know, these processes, once they start, they can take a really long time before we start to see improvements. And I guess my take-home message to you guys tonight is all about making sure that we ensure that we are checking our hives regularly. If you can just switch to my slides now, Susie. Keep in mind that we don't have these pests here yet, and we really don't want them. Although we did get some Varroa jacobsoni in Townsville. At the moment, all is going quite well with that eradication and scheduled for April, so that's some good news. And of course, many of these viruses, we may get them when we get Varroa, and so, as you said, things might look okay for a while. We might get a few years where things don't seem like they're a big problem, and then we might start to see those big losses. So we don't want them here. We don't want the Varroa, we don't want the viruses, and we don't want the hornets, because we will get a huge loss of bees and hives. It'll result in increased effort and cost of management, and of course, for honeybee producers that are using their honey and are treating for the mite, we also get those residues in honey. So there's some good reasons for our beekeepers to try and do what they can to keep these nasty pests out. And the best things that we can do is to try and find the pests as early as possible to give us the very best chance of eradicating them. And so what you can do as a beekeeper is to make sure you're regularly undertaking surveillance. So undertaking a sugar shake or an alcohol wash and a drone uncapping to look for Varroa at least twice a year, but you can do it more often than that. And we're going to have a program up and running soon where we can start getting some beekeepers to report back to us and let us know how their sugar shakes and alcohol washes are going. And any unusual pests that you see, please report them to Biosecurity Queensland. I've got our hotline up there, and that will allow us to come and investigate anything as quickly as possible. And hopefully, when we find things, we can then have a better chance of eradicating them. And so we're going to open the questions now and move on to questions. So if you've got a question for our speaker, make sure you type it in. And we're going to start here with one from Lucas. Lucas, this is one, Stephen, this is one for you. And he's asking, what method did you use for detecting deformed wing virus?
So we would just use the classic PCR. So these are well published now. So we've also used PCR, then what's called next generation sequencing. So they're the standard two methods now for using it. So they're well published, but beekeepers can't do it by simply looking at and diagnose, although they'll tell you they can. You've got to do it using a molecular method.
Excellent. And again, if you think you might have something that you're concerned about, what you can do is send in a sample of bees to Biosecurity Queensland, and we can do tests for these types of things. Again, we hope it's not deformed wing virus, but we'd prefer to be safe than sorry. Got another question here, an anonymous question, and someone just asking, is the Varroa a tick or a mite?
It's a mite.
They're very closely related groups, the ticks and the mites, aren't they?
Yeah.
Thanks. And we've got some comments here. Really enjoyed the talk. That's from Kate, so thanks very much. I've got a question myself, and that is, where do you think things are going from here? Do you think it is trying to rely on that naturally being resistance occurring, or do you think you need a combination?
you're going to have options which other beekeepers didn't have. So some countries have gone down the option where they just said no treatment, and they increased monitoring. They would have to destroy colonies and went through a period of short pain. And it was probably 5 to 6 years it takes until the bees can become adapted to the new to Varroa, and then that's it. You've got over this, and a couple of countries have already done that. So a good example was South Africa, where they just decided no treatment. And yes, sure, they had losses. It's very unusual you don't get losses. The other alternative is to basically go straight for the treatment, effective treatment plans, but then you'll get locked into a system where you've got issues like United States, where it's almost impossible to come out off that because there's just so much reliance on it. It'll depend. One of the biggest problems you have is your feral bee population, because you can't help them in that way. So they will initially be sources of huge numbers of mites, and so this is it's going to be very, very difficult. But I think it needs to be on the table that you are going to have, the bees can get over this with help and time, but it's never been done on a on a massive scale. It's been done in a sort of small country scale, but never in a sort of widespread. But you have two choices: is to treat, or then to really go for the selection. If we'll understand there a lot more, hopefully, by the time you get it. It may be the fact that if the genetics are in queens, which we're trying to look at the moment, there's a couple of teams looking at what carries the